we want to look sci-fi, there are technologies that are currently being developed that basically would allow one to take a delivery system, probably an adenovirus-based system, in which you take PAX6 apart and you basically create a library of different PAX6 genes. So you create a whole bunch of these things. So you take the normal gene and you break it up into 20 to 50 pieces. And then you put each of those pieces into an adenovirus, and then you begin putting those viruses, you inject the viruses into the womb and let them infect the child, with the idea of those infected cells will then correct the deficiencies before or as the cells are being produced. Obviously, we're looking way down the road, but it's a technology, it's an emerging technology that will probably begin seeing some clinical use for some types of disorders. Not PAX6 because it's too complicated, but it's something that I could envision PAX6 being amenable to, down the road. Again, follow up, more?
I know several of you had questions before when I've emailed back and forth with several of you. Does anybody want to have any questions specific to the presentation or not specific to the presentation that they want to ask Dr. Lauderdale?
Hey, Jim, it's Adam Gibgot.
Hey, Adam.
I have just a general question about the state of the science and the community, so to speak. Are you finding there's a greater interest in this field from any of your colleagues, or do you feel like you're kind of one of the only people focusing on this area as it relates to the genetics of PAX6?
No, I'm not the only one. There are a handful of us around the world that are working on this right now. Beth Simpson, who's at the University of British Columbia, is working on one sort of high-end genetic therapy, although not for PAX6, but for other genes which are easier. Excuse me. There are clearly some other people who are working on, a lot of people are working on stem cells. Well, a lot is a relative term. There are probably about, 6, 7 labs right now that are working on different stem cell approaches. The NIH is currently working on an adenovirus delivery approach for certain types of PAX6 mutations. So I would say the interest is growing, but right now my lab is one of the only ones that's kind of doing our shtick. Veronica is the other lab that's doing, that we overlap on some of this, and we have our own areas of interest in others.
Thank you.
Okay, we have a question from Rosalind Fast asking, how does hydration affect the PAX6?
How does hydration affect PAX6? I'm not sure I understand the question.
Rosalind, can you hit the talk button to explain more of your question? Okay, I'm going to move on to the next question from Lacey. Lacey, if there is a confirmed mutation in the PAX6 and the ELP4, is there added concern for additional problems? At original diagnosis, there was little known about ELP4. Now I have heard about Rolandic epilepsy and/or yeast intolerances.
Great question. The region that PAX6 is in, as I said, is gene rich, and so the short answer is yes, there are concerns about additional problems. Let me see if I can quickly get back to my slide here. I went too far up. Okay. So if we go back to this slide, obviously I have ELP4 on this slide. ELP4 is a histone, to genes turn on and off in a more global fashion. We don't know as much about ELP4 as we would like, but certainly, well, there are two parts to this. So, first of all, ELP4 is a gene that controls the expression of a whole slew of other genes in a bunch of cells, that some of which are PAX6 and some of which don't express PAX6. Mutations in PAX6 are usually associated with aniridia and everything that goes with it. Deletions that begin taking out additional genes are going to begin to add on different problems. And so there is some evidence that ELP4 is involved in epilepsy. That data is still in the early stages. It's probable that it's the combination of ELP4 plus PAX6 plus actually something else that's contributing to that. Certainly, if you have a deletion that runs, I'm looking for my genomic slide at the moment. Certainly, if you have mutations that start taking out Wilms tumor or RCN, there are complications with Wilms tumor gene. RCN is involved in neural development, so that brings in epilepsy and mental retardation. Genes to the right side of this locus, I'm sorry, I'm babbling. The short answer is, as soon as you start taking out more than one gene, you're going to start getting additive effects that are going to give rise to new components to the aniridic spectrum. So, let's see, where did it go? So there is additional concern. In the case of ELP4, it's probably not a high concern, but if it gets out to WT1, there is an exceptionally high concern for Wilms tumor. And that's just simply because of the way the genes work. Most people seem to get along quite happily with only one copy of ELP4 and with no changes. So I hope that helps. So Lacey, did that answer that well enough?
And Amy has an add-on to that. Her daughter has a normal PAX6, but a ELP4 came back. Is there any complications that are more or less severe with mutations within PAX?
Amy, so was the mutation in the ELP4 gene, or did it say that there was a deletion? Oh, I'm sorry. I just read the question, so I understand. Okay. So the complications, so the people in our study that have an ELP4 mutation or deletion, they have classic aniridia. So the effects of the deletion are exactly the same as somebody who has a nonsense type mutation in PAX6. And so as of right now, we don't see any additional complications for that. It's no worse or no better.
Thank you.
Okay, and Rosalind she followed up. She's fine. I find when I have facials and eye treatments that they explain how my eyes seep up the serum and I have trouble staying hydrated. Is there a connection between aniridia and underdevelopment of eye parts and staying hydrated?
Yes, most definitely. So dry eye is a very classic, part of the classic aniridic phenotype. And actually, I can show that. So even though this slide is not designed to do this, if you look down where you see Kim and Lauderdale, so right down here, and you can kind of see it right up here at the very top of the slide. So those parts you can see are expressed in PAX6. So those parts of the eye are what give rise to some of the glands that will help keep tears and keep your eyes wet. And so we know for sure that a mutation in PAX6 is going to affect the development of those tear ducts and glands, and so dry eye is most definitely a part of the for many people is a part of the aniridic phenotype. Next question.
We had one question from our Facebook page. Somebody who they don't have a history of aniridia in their family, but 2 of their 3 children has aniridia.
Okay. So clearly this is sporadic aniridia. You guys don't know how surreal this is to be staring at my computer screen talking to it. So clearly this is sporadic aniridia, and I'm going to come up junction spaces. Okay. So if you look here at case 1, usually the way you read this diagram is that dad's a square, and I think that's kind of appropriate, and mom's a circle, and then when you get a shaded box, that means that the child is affected. And so in this case, we didn't realize initially that mom had aniridia because she had a very mild form of it. And so my guess is that one of the parents, even though they haven't been diagnosed, most likely does in fact have aniridia. And if I had to guess, it would probably be mom, although it could be dad. The other possibility is that the mutations occurred in either the germline of either the mom or dad, so that they themselves are not affected in any way, shape, or form, but their offspring now have some frequency or probability of getting aniridia. The fact that there are 2 children in the same family makes me think that either one of the parents is in fact affected and hasn't been diagnosed, or that the mutation occurred early in one of the germline, probably early in the development of either the sperm or the eggs, and so that's where it's coming from.
Thank you. Is there any other questions from anyone in the group? Okay, Dr. Lauderdale, we really, on behalf of Visitor Tomorrow and really the whole aniridia community, thank you for giving us hope through your work. We hope to continue to support you, and if there's anything that we can do, please continue to let us know.
Thank you. But before we sign off, I'm really curious how many people found this useful. Don't tell me no, I guess, but you could hit a check mark or something. Or I guess I should ask, does anybody didn't find it helpful, and tell me what I can do to improve? That would be good too. But thank you all for taking the time out of your days to come listen to this, and we'll do it again.
Thank you, everyone. Thank you, Dr. Lauderdale.
Goodbye.